Good morning, everyone. This is not the time to stress. This is time for you to do your best. All that you have learned with me, I had done the recaps, whiteboards, reviews in advance so that you could prepare in the week before the test. So you have all the information. Please see every help file is under the exam one folder. What I'm going to do is refresh you. Right? As you know, there are 20 questions. So let's start our recap. As you know, there are 20 questions. There will be 3 questions from overview, 5 from enzymes, 5 from ETC, 5 from signal transduction, and 2 from carbohydrate digestion. Let me okay, carbohydrate digestion. Now let's see how I'm going to do this recap. I'm not going to teach here. I'm going to go over this, and in the end, I'll give you about 5 minutes to ask questions. Please, I have divided this recap into 2 phases. First phase, we will do overview, enzymes, and carbohydrate digestion. I'll give you a 5 minutes break, and in the second phase, we will do ETC and signal transduction. Please know that I was invited for this NBOME exam item writing workshop. And so I don't choose the date and time. I'm glad I did it, but I hope I helped everyone and everybody's good. So let's do that recap. And metabolic map will be given on your test. Metabolic map is more beneficial in foundation too, because there we start metabolism. What did we do first? Catabolism versus anabolism. We try to distinguish that, saying that catabolism gives you acetyl-CoA, which is a source of, so quickly, if you can also participate in the chat. So catabolism gives you acetyl-CoA, which is a source of carbon or hydrogen. Acetyl-CoA. It's a source of carbon. Catabolism also give you NADH, which is a source of ATP, NADPH, which is a source of hydrogen. Now, all that from catabolism is required for anabolism. But there is one hormone which is mainly anabolic. Anabolic to what? Lipid and protein. Insulin also kicks in. So yes, catabolism gives you the requirements of anabolism, and we have clearly distinguished catabolism from anabolism. You know very well. One thing you cannot do anabolism without is what? That's the universal requirement for anabolism. ATP, absolutely wonderful. Then we identified catabolism, which is an oxidation reaction. It is exergonic. The energy released is also known as Gibbs-free energy. It's a useful form of energy. Now, sometimes students are confused. Delta G naught, delta G naught hash, they're all the same thing. They stand for Gibbs-free energy, which is a useful form of energy because oxidation of foodstuff release that energy and we store them as ATP. So yes, we also identify as catabolism as oxidation, exergonic reaction, and the energy release useful trapped as ATP. We did talk about the big oxidation reaction as oxidation accompanied with reduction. And that's what I'm going to also pinpoint when we are doing electron transport chain. Just like oxidation in our body is something gets oxidized and the next component gets reduced. It's same with ETC. So did we talk about a big enzyme, oxidoreductase? And what was the subclass of this oxidoreductase? Which was used in catabolism and oxidation. Big subclass, dehydrogenase. Then we also identified that these enzymes cannot function alone. They need vitamin and coenzymes. So I'm just trying to pinpoint what are the different, I'm just trying to pinpoint where and how you got to connect, right? I'm not teaching here. So have we addressed all that? So yes, oxidoreductase was a big class, subclass dehydrogenase involved in catabolism, which is not oxidation alone, but oxidation accompanied with reduction. But enzymes then are helped by vitamins, which act as coenzyme. We talk about them? Absolutely. Then we did the multi-step enzyme catalyzed pathway. Now, some students are confused who is subjected to regulation. Well, irreversible steps are subjected to regulation. Irreversible steps like E1 and E3 can be rate limiting or committed, but they are subjected to two types of regulation. And what does short term do? Short term and alters enzyme activity. So it's not that only rate limiting step or committed step undergoes regulation. All the irreversible step, whether they are rate limiting or committed, there is no specification on that they can undergo any regulation. So which took days and was at the level of gene? Was it long term or short term? Long term, it was at the level of gene, and it took days. Short term, students get confused. Although short term is in minutes, but it's all reversible, right? It's for regulation, so it's reversible. But did we talk about glucokinase? So where is glucokinase releasing insulin from? Where? Where is glucokinase releasing insulin from? You remember glucokinase release insulin. Glucokinase releases insulin from pancreas, right? And when do you want to release insulin? After meals or right when you wake up in the morning? After meals. So that's why now you can understand and what enzyme you have in. So glucokinase, you clearly identified, helps releasing a pancreas after bake sales. Yes, absolutely wonderful. I'm glad you remember bake sales for carbohydrate digestion. So absolutely wonderful that you have glucokinase in the pancreas releasing insulin. That's why. And where do you release insulin? After bake sales. So will glucokinase be active when you wake up or after meals? When you wake up in the morning, after meals. So this shows that glucokinase is under substrate concentration. And that's how we distinguish glucokinase and hexokinase. Glucokinase will never be active after fasting, right? This students are confused with. It's also a type of short-term regulation, reversible. And have we done a reversible covalent modification and allostric? So which one of them shows S-shaped curve? The reversible covalent modification or that allostric regulation, which is positive and negative? So allostric regulation is different from reversible covalent modification. Absolutely wonderful. You said long-term regulation take days and work at the level of gene. Sorry, if we go back, it take days and work at the level of gene. Very good. And short-term regulation is reversible, is quick. And look at glucokinase. It should be active after meals, not when you wake up. Look at this one when you talk about reversible covalent modification. So now, how do you get good grades when you correct one lecture from the other? For example, have you identified that insulin docks different proteins for its diverse role? Does insulin dock different protein for its diverse roles? Did you learn that? Yes, one of the protein can be this too, which is not in the figure of Lippincott or Marx, which is in my lecture slide. It can also dock this protein. So does insulin always dephosphorylates the key enzyme? Does insulin always dephosphorylase the key enzyme? But now you can add mechanistically. First of all, you should know what is insulin docking to do this action. Now, when insulin docks protein phosphatase, will it do growth factor-like activity? If insulin docks this protein phosphatase, will it do growth factor-like activity? Yes or no? No, no, because insulin has to dock which protein to do growth factor-like activity? Not GRB2. Very good. So see, insulin has specific protein to do the specific outcome. Absolutely wonderful. Insulin has to dock GRB2 to do growth factor-like activity, not protein phosphatase. Protein phosphatase is when insulin will dock, it will dephosphorylate the key enzyme. So there are different protein, different outcome. Then we did enzymes. They are protein in nature. We identify the non-protein part. And vitamin act as, you can't forget your daily vitamins and vitamins for the test too. Vitamins act as coenzyme or coenzyme. Absolutely wonderful. This is overall Gibbs free energy change, which is released by catabolism, oxidation, useful form of energy. Is it altered by the enzyme? Yes or no? Is overall Gibbs-free energy altered by the enzyme? No. Did we do this big class oxidoreductase and a subclass dehydrogenase? What is it involved in? Is it involved in oxidation in your body? Oxidoreductase and the subclass dehydrogenase? Yes. Is it involved in oxidation in your body? Oxidoreductase, and NAD is used more than FAD. Then we'll go to electron transport chain in the second hour. Now, see, we also did clinical enzyme. Why did we do them? Because they tell you specific tissue location. For example, alkaline phosphatase. Is it if it's rising in your circulation above a normal level, does it tell you bone damage or prostrate? Does it tell you pathology is in the bone or prostrate? If alkaline phosphatase is rising, bone, very specific. We also did isoenzyme. Then graphs. Become comfortable with graphs. But before doing comfortable with graphs and getting to inhibition, know this is velocity versus substrate. Know the shape of the curve. Know what's Vmax. So when Vmax is reached, is the reaction at saturation? Yes. Yes, it has saturation. Can Vmax be altered by altering the substrate concentration? Can Vmax? No. So trying to understand what is V, what is Vmax by 2, what is Km? Km is on the x-axis. Km reflects affinity or efficiency of the enzyme. Affinity. So understanding these terms. Yes, initial part of the reaction, there's a proportional change. This is first order. Later part of the reaction, it is saturation. What is saturated? Enzymes. When you're saturated, you reach Vmax. Can you alter Vmax by altering substrate concentration? No. Then this hyperbolic curve moving right, left, up, and down. This hyperbolic curve moving right is what? Activation or inhibition? Moving right, this hyperbolic curve. Moving right is inhibition. And if you're moving right, what are you changing? Km or Vmax? What are you changing? Km. When you are moving right, are you increasing Km or you're decreasing Km? When you're moving right, you're increasing Km. And when you are increasing Km, is it competitive or non-competitive inhibition? Competitive inhibition. Absolutely wonderful. So see, you understand those terms, what they reflect for. Now you go to this line weaver book. I hope you're following my pace. Yes, because I want to just refresh these 3 lectures. Absolutely wonderful. Great. But when you do 1 over B by 1 over S, it was a straight line extrapolated back. So did you have an x-intercept and a y-intercept? So which one reflects Vmax, x-intercept or y-intercept? Y-intercept, absolutely wonderful. You have to be clear with that. Then when you move this straight line right, is Km increasing? When you move the straight line right, is Km increasing? Is Km increasing? Absolutely wonderful. Where does Km increases? Competitive or non-competitive inhibition? So I'm explaining you one, competitive inhibition. Very good. When this straight line moves up, is Vmax increasing or decreasing when the straight line moves up? Decreasing. Where is Vmax decreased? Competitive or non-competitive inhibition? Non-competitive inhibition. Now somebody asked me, I like your questions. Your students are very proactive, great questions. In line weaver work plot, I said that inhibitor line should always be above the control line. For line weaver work plot, inhibitor line should always be above the control line, not below the control line. So see, I asked you to what's Vmax, what's Km, what do they reflect? Then we went to hexokinase and glucokinase to differentiate it. Right? You also did your transporters. Dr. Pinstock has beautifully explained all of them. SGLT1, SGLT2, GLUT1 and 3, 4, and 2, right? She has explained the transporters. And see, again, connecting better grades. Connecting them gives you better grade. Like these, remember that yes, we have a few questions to ask. So they will be second and third order. But don't forget, if your basics are clear, there's no flip-flop there, right? Don't wear flip-flops on the exam, right? I'm just joking. So if there are no flip-flops there, your application can never go wrong, please. So just hammer those points. Don't flip-flop them and you will be fine. Yes, we identify this first order also done by GLUT2. Where is GLUT2? Brain RBC, liver pancreas. GLUT2 brain RBC or liver pancreas? Liver pancreas. GLUT2 pancreas, you need a proportional flow or flow all the time. GLUT2 pancreas, you need a proportional flow. That's why they follow first order kinetics. Just like enzyme. Zero order was 1 and 3. Zero order means saturation kinetics, constant kinetics. Who also follows that? 1 and 3. Where? Where is 1 and 3? Most peripheral tissue also in brain and RBC. You want a constant flow. Here, the same concept, though you extrapolated, that when you reach Vmax, it is independent of substrate concentration. So can you alter Vmax by altering substrate concentration? Can you alter Vmax by? No, you can't. And then you go to whether this hyperbolic curve is moving right, left, up, or down. You can't flip-flop that. Moving right is not activation. Hyperbolic curve moving right is what? Hyperbolic curve moving right is, so you got to get first comfortable with these graphs. Hyperbolic curve moving right is inhibition. Absolutely wonderful. Then cam, what is low cam? What is high cam? Low cam shows high affinity. Is that true? Absolutely wonderful. Then we did hexokinase and glucokinase, which people are always confused about. Please know that these, I have too many pop-ups, but I'll try. So I'm just watching your chat too. So there are don't get panicked with what is glucokinase, hexokinase. Uh, Understand that glucokinase is in pancreas. And what is pancreas for? To secrete. What is pancreas for? To secrete what? What's your final goal? To secrete insulin. So you need an enzyme of high efficiency here. High efficiency is reflected by Vmax or Km. High efficiency is reflected by Vmax or Km. That's why glucokinase is an enzyme of high Vmax. Whereas hexokinase is in brain and RBC, where you want to quickly utilize glucose. So hexokinase should be enzyme of high affinity. What is high affinity reflected by? Low Kms. That's why hexokinase has a low Km and rightly there. Also, please, first there are transporters on the surface, then the enzyme is inside, which has different kinetics. You're not doing carbohydrate utilization right now. You're being very mechanistic that, okay, this is my transporter, this is my enzyme, what is its kinetics? We are still at a very basic level. And then we will connect in foundation two. So transporters are on the surface, which push in, and enzyme is inside for the outcome. So proportional flow, enzyme of high efficiency. Are you clear with that? Are you clear that you need an enzyme of high efficiency in liver and pancreas? That's why rightly, which enzyme is there of high efficiency in liver and pancreas, especially pancreas, because you want to secrete insulin? Glucokinase. So you also understand hexokinase, glucokinase there with kinetics. Then you come to inhibition. Students are confused with this covalent. Covalent is mentioned only twice in your lectures. One is with irreversible and one with reversible covalent modification. Covalent only means strong. So don't make it similar to irreversible, just strong. Because covalent was used in reversible covalent modification. What was reversible covalent modification? Was it long-term or short-term regulation? Reversible covalent or short-term regulation. There also I used covalent, but there it was reversible. Here I'm using covalent under irreversible inhibition. So covalent just means strong. Do not confuse it with reversible or irreversible. Then we clearly distinguish competitive from non-competitive inhibition. Also with hyperbolic curve, I told you, see what is changed. With Line-Weaver-Burg plot, see what is unchanged. So what has changed here, Km or Vmax? What has changed here, Km or Vmax? Km is changed. Is Km increasing? Is Km increasing here? Yes, and where does Km increase? Is competitive or non-competitive inhibition? Competitive inhibition. If you want to understand why, yes, because why Km is increased in non-competitive inhibition? Number one, The affinity of the enzyme for substrate decreases in the presence of competitive inhibitor because they compete for the same site. So when the affinity is decreased, what is increased? When the affinity is decreased, what is increased? Km. Also, you can understand why Km increases in competitive inhibition. You can overcome this inhibition by increasing substrate concentration. So when you increase substrate concentration, what increases? If you increase substrate concentration, what increases? Km or Vmax? Km again. So you can understand also why Km is increasing, but finally you got to get to comfortable with the graphs. So in a hyperbolic curve, quickly see what is changing. What is changing here? Km or Vmax? What's changing here? Km or Vmax? Km. Is Km increasing? Is Km increasing? Where does Km increases? Competitive or non-competitive? Competitive inhibition. In a line Weaver plot, see what is unchanged. So is the x-intercept unchanged or y-intercept unchanged? What is unchanged? Y-intercept. What does y-intercept reflects? Km or Vmax? Vmax. Where is Vmax unchanged? Where is Vmax unchanged? Competitive inhibition. But in a line Weaver plot, inhibitor line will always be above the control line. Green is the control line and blue are inhibitor concentration increasing. So I said that inhibitor line will always be above the control line for the line Weaver plot. Green is the control, green here is control, and blue is increasing competitive inhibitor concentration. Also, please understand that inhibitor binds to the enzyme or the substrate. Where does inhibitor bind? Inhibitor binds to the enzyme and changes the kinetics of the enzyme. So you should be very careful of the basics. Yes, there's a change in kinetic of the enzyme, not the substrate, not the coenzyme. There's a change in the kinetic of the enzyme in the presence of competitive or non-competitive inhibitor. That's why there are different graphs. Do the same for line Weaver plot and hyperbolic curve for non-competitive inhibition. So what do you see changing here? Okay, y-intercept or x-intercept? What is unchanged here? X-intercept. And always inhibitor line is above the control line. I have explained you competitive. You could do the same with non-competitive. In hyperbolic curves, see what is changed. In line Weaver plots, see what is unchanged. If you want to understand with kinetics, I can remind me, I'll do it in the end so that I first do everything, right? I will also tell you how you should know by kinetics. Some people don't memorize. They want to know why Km is increased. I'll quickly tell you. So see, first you get comfortable with a graph. First, see the variables always. It's between V versus S. Then see the shape. It's hyperbolic. So when it's a hyperbolic curve, is it allostric or non-allostric? Hyperbolic means non-allostric. Do they follow Menton's kinetics? Do non-elastic? Yes. Do they have a KM? Do they have a KM? Yes, they have a KM. Then you see what is Vmax? What is KM? KM is the substrate concentration. Then what do they reflect? What does KM reflect? What does Vmax reflect? Have we done hexokinase, glucokinase with their respect? Yes, you have clearly done that. Then is this curve moving right, left, up, or down? Does that make sense? Where it is moving? Clearly, you should understand. Yes, competitive inhibitors, although they don't have change of kinetics, please know that. So an inhibitor binds to the enzyme and changes the kinetic of the enzyme. So yes, both in the presence of competitive as well as non-competitive inhibitor, there is change in kinetics of the enzyme. In competitive inhibitor, what's the change in kinetic of the enzyme? It's Km. In the presence of competitive inhibitor, Km increases. What happens with the non-competitive inhibitor? How does it change the kinetics of the enzyme? Vmax decreases. So it's the inhibitor whose presence changes the kinetics of the enzyme. Yes, if I answered you correctly. Very good. You should also ask questions. This is what I will open last 5-10 minutes for that before we take a break. Again, You should get comfortable with the graphs. Hyperbolic curve, you see what is changed. Double reciprocal block, you see what is unchanged. And why I will address at the end. Let me finish the slides. Irreversible or suicidal should look like competitive or non-competitive. Irreversible or suicidal should look like non-competitive because they are also binding to the enzyme and decreasing enzyme concentration. S-shaped curve. S-shaped curve is for elastic or non-elastic. Again, you see graph, don't panic. Velocity versus substrate. S-shaped curve, this one. This is for elastic enzyme. Do they follow Manton kinetics? No. Do they have a Km? No. Are they K or V type? Yes. So elastic have enzymes have properties. That's why they are known as elastic enzyme. How can you quickly identify them from the graph? Shape. They do not follow Manton's kinetics. Yes, S-shaped, absolutely wonderful. Then they are K or V type. If they move the curve right or left, K type, up or down, V type. So tell me, moving the curve left and up, is it activation? Moving the S-shaped curve left or up, is it activation or inhibition? It's activation, right? And when I say that inhibitor line is above the control line, that is for Line-Weaver-Burg plot, right? Now comes the last section, which is your, for the first 1 hour recap, is your carbohydrate digestion. Please, I always encourage students to watch Dr. Binstock's slides, the professor slide. Only thing, I'm giving you a skeleton. So what's an overall skeleton for carbohydrate digestion? First, you got to go to bake sales and consume them, right? So starch you get from plant, glycogen you get from animal, milk, lactose, and table sugar, sucrose. Is glucose found free in nature? No. Fructose is found free in nature. Where do you get fructose from? Where do you get fructose from? Fruit juices and honey. From bake sales, you collected them all. Monosaccharides are not just the best reducing sugar. Please don't do this. Remember, this will come in foundation too. But I want you to know that monosaccharides are not just the best reducing sugar. That's why they give dipstick tests positive, but they are also the only and the only absorbable form. Why? Why monosaccharides are the only absorbable form? Why does body do that? What's the reason? That monosaccharides are the only absorbable form. Anyone? Because they are less osmotically active and can be easily absorbed. Absolutely wonderful. So you went to bake sales, then you digested carbs in the mouth, very less transit, pancreatic amylase, salivary as well as pancreatic amylase, not responsible for complete digestion. Yes, you have other enzyme and disaccharidases in the intestine, which are responsible for complete digestion. And what is the complete digestion and the only absorbable form? Monosaccharides. And this is how, so this is how you got to link so that you don't go wrong in foundation 2 too. Remember, every lectures is designed that the foundation 1 help should give you get good grades in foundation 2, although they are not retested. But believe me, once you do your each foundation exam well, your other exams are linked to them, so you will always do better. And I'm sure you all will do well. So this is the sequence. You collect all your sugar. Why is sources important? For example, if somebody is having, if somebody is having an aversion to fruit juices, will you limit lactose or sucrose? For example, if somebody is having aversion to, you will limit sucrose, not lactose. So how would you know that? Fruit juices means it is coming from, so sources is important. Fruit juices means it is coming from where? Which sugar? Fructose. And fructose is a part of lactose or sucrose? Fructose is a part of lactose sucrose. So understand? So you knowing your sugar, what are they made up of, their sources, is important for you as a clinician to also advise diet and know which diet will cause signs and symptoms. So you want to make big sales, you collected all the carbs, I told you the sources, you had amylases and disaccharidases. Finally, in the intestine, sugars are ready for absorption. They move from lumen to cell, cell to circulation, and then they are picked up by tissue. Somebody was asking me, where are these transporters? These transporters are on the luminal side or on the contraluminal side, but peripheral tissue, they are on their surface. As transporters do, they will transport the whatever their function is to transport. So there are sugars that move monosaccharide from lumen. Sorry, there are transporters that move monosaccharide from lumen to cell. There are transporters that move monosaccharide from cell to circulation, and then there are transporters that pick them on peripheral tissue. Dr. Binstock has explained all the transporters. You know SGLT1 is facilitated diffusion or secondary active transport. SGLT1, secondary active transport. Do you also have SGLT2? Where is SGLT2? Where is it's in the kidney tubules. Very good. So there are specific transporters, they have specific kinetics, right? But once glucose is in the circulation, which is your predominant sugar, they can be picked up by tissues. And this is the sequence: bake sale, amylases, disaccharidases. Can you name one disaccharidases, please? Any one disaccharidases you want to name? Lactase. So disaccharide lactase is responsible for final digestion. Monosaccharides are ready for absorption. The transporters do all the transporters. I have not mentioned SGLT2 here, but it's in the kidney tubules. So you go back to Dr. Binstock slides and do them. So then there are transporters that move them from lumen to cell, cell to the circulation. And now remember your sequence. Who has the proportional flow? Since it's after meals, where will glucose flow now? Proportional. Where will GLUT2? What will pancreas do? What will pancreas do now? After meals, what will pancreas do? Glucokinase GLUT2 release insulin. And which transporters are insulin mediated? That will be GLUT4. They will be expressed. 1 and 3 have constant flow. They don't need insulin. GLUT2 have proportional flow. They don't need insulin. Who needs insulin? GLUT4. Why are you doing that? Well, you want sugar to be used now. You're going to bake sale, you collect glucose, you want to push all the sugar inside the cell and turn on its oxidation. But believe me, if you didn't have different transporters first, so that's why please go basic. If you didn't have different transporters, can you start glucose utilization inside these cells, these peripheral tissue? Can you even start glucose utilization? No. So foundation 2 will now tell you how to do glucose utilization. So don't go there. Go basic. This is my source. This is my transporters. This is how their kinetics is. Then when I come back in foundation 2, I will connect. You're not also doing this mechanism of insulin release, but yes, you have identified GLUT2 proportional flow. You have identified difference between glucokinase and hexokinase. You clearly know that glucokinase is responsible for release of insulin. We are not doing mechanism, please. We are not doing mechanism. I have not done mechanism. And insulin is going to do what? Push all the glucose inside the tissue. How is insulin achieving that? How is insulin pushing glucose inside the peripheral tissue? What do you need? How is insulin pushing glucose inside the peripheral tissue? No, you need transporters, whether GLUT4 is the only one. Very good. I like that answer. GLUT4 is the only transporter which is insulin mediated. So insulin is increasing GLUT4. But others have their own flow. But then, is insulin an anabolic hormone? So then have we addressed anabolism and catabolism? Have we addressed anabolism and we have identified insulin as the anabolic hormone? Very good. So do anabolism and catabolism. Go very basic. We are not doing all this. This will come in, the carbo pathways will come foundation 2. So don't break your head here. Also, have we done that once you go to bake sales, you collect your sugars are moved from lumen to cell, cell to circulation by transporters. Is GLUT2 proportional, and is glucokinase enzyme of high Vmax releasing insulin? And when you release insulin, how is insulin going to act? That will be, I'll do in the second phase. Then you come to their signal transduction mechanism. So all basics, you're not doing these pathways, please. All you are doing, trying to do is basic transporters, mechanism. Okay. Where are they? Which are insulin mediated? Which are proportional? Which has constant flow? Basics, don't do metabolism. You know, lactose should be digested by which enzyme? Which enzyme should digest lactose? Where? Where? Where this lactose should be digested by lactase? Small intestine. Very good. Yes, lactase are attached to the brush border of epithelium, intestinal epithelium. That is why you have secondary lactose intolerance. But if it is not digested in the large intestine, is it good to have a disaccharide? Is it good to have disaccharide? No, it will be osmotically active. It will draw in water, watery diarrhea. Fermentation product will cause cramp and pain. And that is why one of the fermentation product is also known as a non-invasive hydrogen red test is used for diagnosing lactose intolerance. So it should be digested where? It should be digested where? Small or large intestine? Small intestine. If it is in the large intestine and you see lactose has not been digested, then that's not correct. That's not good. It will create a havoc. Although it is only bloating, pain, cramps, not floating. It's just a presentation. So don't think we are floating with lactose intolerance, please. And that's all for the first recap. I'll give you a break and you can ask me questions and we can start at, you can join me back at 11. Then we will do, if you have questions, you can ask me now. Then at 11, we will do only ETC and signal transduction. Great. Any questions? Otherwise, if no one has question, then maybe we can take a 5 minutes break and I can no, but I think other students know that we start at 11. So I would leave it for question answers. Sorry. Yes, Noah. Great.
Hi, Dr.
Okay.
Bog. I just had a quick question about uh, GLUT 1 and 3. In some of the
Yes.
example questions, I noticed that you included GLUT 3 as being on the brain in RBCs, but I'd
It's
also seen
most
that GLUT
peripheral
3 was
tissue.
just on the neurons. So I just want to see if on the test, if you were going to delineate
No,
between
no,
GLUT
no,
1 for
because
brain.
no, because see, my idea of distinguishing GLUT2 with glucokinase is because though you have done enzyme kinetics, but transporters go with them. So I want you to relate GLUT2 with liver and pancreas, and GLUT1/3 is with brain and RBC and most of the peripheral tissue, but not specifically now. That will come in foundation too. But I want you to connect them both at these two different tissues. Does that make sense? Rather than being specific? Did I
Yes, no,
answer?
it does. It does. Thank you very much.
Right. Can you explain reversible covalent modification? Let's go to the slide. It's more visual. I'm sorry. Is it good to open it for question and answer? Everybody is good like that? Then we will restart at 11. Yeah, let's do what somebody has reversible covalent modification. So no, it was your question, Noah. Did I answer your question? So you just wanted to know that is it that specifically you should know where GLUT1 and 3 are? Am I right? Was it Noah? Was it Noah? I don't know. Somebody asked GLUT1 and 3, sorry. So
That was me, yeah, but you answered my question.
okay, great, great. No specificity, please. Actually, I have introduced them in extra because I want you to connect them both. But basically, you should go back to your glucokinase, hexokinase, right? And somebody's asking this question. Let's see. Visual impression is always good. First of all, it's a short-term regulation, which is reversible, take minutes, right? Covalent modification is only you are seeing, as you see here, there's nothing. It's a type of, you have to go mechanistically in this exam, right? Basics, mechanism, then clinical. Don't forget clinical. I'm not saying you're not being tested on clinical. So this is just phosphorylating and dephosphorylating the key enzyme. You need something to phosphorylase. Also, some students get confused. I'm just telling you, anytime you have to add a phosphate, what enzyme should come in? Anytime you have to add a phosphate, there's only one, kinase. So again, you need a kinase to add the phosphate, and you need a phosphatase to remove the phosphate, because you will know regulation in foundation too that you want to sometimes phosphorylate. After function is done, dephosphorylate the key enzyme. So that's all reversible covalent modification. Why it's called covalent? Because phosphate is attached covalently, but it's just adding phosphate to the key enzyme, removing phosphate from the key enzyme. That's reversible covalent modification. And if you connect this protein kinase, is which one for glucagon and epinephrine? Is it A, B, C, or G, this protein kinase? Do you remember? This is A, right? So see, if you connect your metabolism with signal transduction, that will also help you. Rather than, okay, glucagon and epinephrine is phosphorylating, but okay, what was here, right? Did I talk about this though? Insulin can dock this protein? Can insulin dock this protein? Yes. So that is reversible covalent modification. Glucagon and epinephrine will activate pKa to do that, and insulin will dock protein phosphatase to do that. That's all. Did I answer your question?
Yeah, thank you.
Great, great. What else? Yes, sure. This is your chance to ask question. Then I'll give you a 5 minutes break. I would like to answer your question. Signal transduction is at 11 because I have told clearly on the announcement I don't want to start it early. Signal transduction and ETC will start 11. Does that make sense, Diego? I'm just opening this for question answer. It's just I don't want other students to miss it, right? Does that make sense? K-type and V-type. Now, K-type and V-type is for allostric enzymes, right? Yes? Because allostric enzyme do not have a CAM, but yes, just like any other inhibition, are the inhibitors altering? Do the inhibitors attack on the enzyme, are attached to the enzyme and alter their kinetics? No, you don't need to know structures. There's no structure, chemistry. No, we are not doing that. This is biochemistry applicable to clinical medicine, please. But yes, you should know what your sugars are made up of, just as I told you, right? So, yes, did I answer you? No structures, please. Did I answer you? Okay. Yes. Yes. Should we know alpha and beta? No, no bonds, no structure. Primers are not on the exam, please, either for DO or MS. Okay, I will explain you. Can I finish somebody's? I will explain you what is intense regulation. I will explain you, but let me finish this. So let's see a allostric enzyme. First of all, how do you identify? By the shape of the curve, as shaped. They do not have a KM, but as I told you, you should get used to this, that the negative modifier binds to the enzyme and alters the kinetic of the enzyme. So how is that then that V or K type for allostric? If the S-shaped curve is moved right or left, it's K-type or V-type? S-shaped curve move right or left, is it K-type or V-type? K-type. And an S-shaped curve move up and down is V-type. That's how they are labeled as K or V-type. But it's the same thing, that the kinetics of the enzymes is altered in the presence of inhibitor. Did I answer you?
Yeah,
Yeah.
I was just wondering why, like, what K type means, like, why it's not a KM.
No,
What is
because
it
they do
represent?
not have a KN. Allostric
There's no
enzymes.
affinity?
No, allostric enzymes do not have a KM, and they do not follow Manton's kinetics. So you will not extrapolate that hyperbolic curve kinetics to S-shaped enzyme. They are different enzymes. It's just K-type and V-type. That doesn't mean if you move right, that is activation. It's still the same. If you are moving right, it's inhibition. If you're moving left, it's activation. If you're moving up, it is activation. And if you're moving down, it's inhibition. It's K
Okay.
and V type depending on how it's altering the kinetics of the S-shaped curve. Was it moving right, left, up, or down?
Okay, thank you.
Right? But they do not have a KM. Yes, somebody said regulation okay. So what is your question actually, please? As I told you, there are multi-step enzyme catalyzed pathway. Some steps are irreversible. They could be rate limiting or committed, but they can be subjected to any kind of regulation. But what is your question about that? If you want to clear,
Um,
yeah, sure.
Hi, Dr. Buck. There was a quiz question that basically said a committed step in the multi-step enzyme catalyzed pathway, and it gave like 5 different options.
So
And
either it
at
should
first I
be.
thought it was the intense regulation, but the correct answer was the early in a linear pathway.
Yes, absolutely. That's true that if you want to distinguish committed from rate limiting step, committed step should be early linear. This is the key. And rate limiting step is slow with high activation energy. So that means slow and intense. When you say slow intense, it is for rate limiting step. So does that clarify?
Yes, that makes sense. Thank you.
Yeah, and also when you say actually rate limiting, why you say slow and intense? When I told you delta G negative, is it spontaneous? Delta G negative, is it spontaneous? Will it go to completion? Yes?
Yes.
So the same delta G negative is if it's of higher magnitude, that becomes rate limiting. That's why slow and intense is for rate limiting, and early and linear is for committed. That's true, if I clarified that. Yes.
Yes, thank you.
Great. What else? Your question? Any questions on on overview, enzyme, or carbohydrate digestion? I hope this recap helped everyone. Yes. So do you want to? Sorry. I said elevated enzyme because remember these enzymes are within the cell. Whenever there is a breach in cell lining, these enzymes will move from cell to circulation. That's why high levels of these enzyme above the normal level in circulation don't always reflect some kind of specific tissue damage. That's where I connected that if alkaline phosphatase is high, then the damage is bone or prostate. So it is specific. If alkaline phosphatase is rising above the normal level in the circulation, is it the source is bone or prostrate?
Bone.
Alkaline phosphatase. Sorry?
Bone.
Yeah, bone. So that is what when I talked about specificity. We also did isoenzyme with the same specificity. Please see, I'm bringing in concepts which can have a problem. You can go back. You have also done isoenzyme with the same uh, concept. I want to hammer concepts. Then you can, I can't bring all the slides. Does that make sense?
Yes. Yeah. Thank you.
Okay. Can we take you want to take a break and then come back? Is that working? I thought I just don't want to start before 11, please, because everybody knows signal transduction and ETC is 11. Okay, then let's start at 11. Take care. I'm here. Okay, everyone. Sure. We will start in about 2 minutes, right? Let's get back. And we will do now this half. We are going to do ETC and signal transduction. And let's see how we can bring it back together. And I'll again leave last 10 minutes for question. Okay, everyone? Yes, yes. Sure, I'm just noting down everything in the chat. In the end, I will address everything. Don't worry. This is also for your questions and concerns, right? Let me know when it's 11 because I want everyone to benefit, right? I think 1 more minute. And please see that I have got the slides so that, I can talk about concepts that are really students are struggling with or I have more questions about. You already have so I have posted your detailed recap and mini recap and also all the topic list. And you have your interactive sessions that has the focus, your online practice, everywhere you will see the focus. There is no plethora of focus. Okay, 11. First of all, please see that I went to a nutritional item writing workshop. And unfortunately, I couldn't choose that because I want to bring nutrition in our program. Everywhere, all medical schools are doing it. So I had to go away. And also just to let you know, my daughter is moving to Bing, so I'm here to settle her and say goodbye to her. And I think I made sure that I come back a day early so I could do the review with you all and then say goodbye to her. Okay, thank you. So let's see how, great. Let's see how we can revamp ETC and signal transduction. Please don't stress. This is not the time to stress. You people have really worked hard. I've seen you all were very proactive, whether it was interactive session during my office hours. So let's see how we can go back and redo ETC and signal transduction. First of all, remember that I told you that oxidation, you do catabolism by oxidation, which is hexagonic reaction accompanied by release of free energy. That's useful form of energy known as Gibbs free energy, energy difference in the energy of product to that of reactant and is not altered by the enzyme. We are going to connect that catabolism, oxidation, hexagonic energy, because we know that when this energy is released, so how is this energy released initially in the form of 2H plus an electron? So we know that these 2H plus an electron has all the energy of the substrate, but we now got to trap it in the form of ATP. That's why we need electron transport chain. But don't forget that along with electron transport chain, what do you need to form ATP? Just electron transport chain is enough? Or you need something else to form ATP? What else is essential? There are 3 components essential. You need electron transport chain, mitochondria. There are substrate, but I'm talking about very essential components. Absolutely right. You need the substrate, you need the enzyme, you need the coenzyme. But what are the 3 components, the key, the basic components? You need electron transport chain, you need oxygen, and you need mitochondria, right? Yes? Yes, everything else will come then, whether it's proton gradient, proton circuit, but you need these 3 basic things to form ATP. You need the electron major, I'm talking about major. You need the ETC to be functional. You need the oxygen, good oxygen, and mitochondria. So you are doing catabolism. You're oxidizing food stuff. Energy is released in the form of 2H plus an electron. This is the useful form of energy in these 2H plus an electron, which should go where? To the electron transport chain to synthesize ATP. So this is what I was talking. The key is you need ETC oxygen and mitochondria to do ATP synthesis. Also, are you aware of the word oxidative phosphorylation? That means you're coupling oxidation with phosphorylation. Don't forget, you need a lot of enzymes that everyone is adding to. Coenzymes. Is NAD used more than FAD? Is coenzyme NAD used more than FAD? Yes. And where do these reducing equivalent go? To the electron transport chain to synthesize ATP. Yes, NAD is used more than FAD. Did we do that? All I'm trying to say, you're not wrong when you're saying you need enzyme, as I said, oxidoreductase. You need coenzyme, NAD, which will get reduced after oxidation. You need proton circuit, proton gradient, yes, but the three main pillars are you need ETC, oxygen, and mitochondria to do oxidative phosphorylation. I want you to be very clear with that, so in Foundation 2, you don't mess up. And who's the major pathway doing oxidation? Your TCA cycle, which you don't have to do. But after oxidation, do you get a lot of reduced coenzyme in the matrix? Because the major common pathway is in the mitochondrial matrix. So after oxidation, do you have a lot of reduced coenzyme A ready in the matrix? Yes. Are they ready in the matrix? Which is more, NADH plus H plus or FADH2? Which are more in the matrix after oxidation? So if you have done good oxidation, NADH plus H plus will be more than FADH2. And look at the electron inner membrane has then now all the ETC components where these NADH and H plus and FADH2 will pass their electrons and finally reduce oxygen to water. So who is the ATP synthesis? Which is not a ATP synthesis site? Which is not a ATP synthesis site too. So see, they have given all the complexes. ATP complex 2 is not a ATP synthesis site. Very good. But who synthesizes ATP? Which complex? What is this complex also known as? Complex 5. Is there another name for this complex? F0, F1 complex. Absolutely wonderful. So see how I am going. I'm trying to connect first. Oxidation accompanied by release of energy, useful form of energy. How is it trapped now? In the form of 2H plus and electrons. Where is it? Mainly in the mitochondrial matrix. Where do they have to pass them? To the electron transport chain. So you are going normal, then you do inhibitor. So first you need to understand that there are 5 fixed complexes clearly shown here and 2 mobile carriers. Then you should know, are more substrate linked to NAD than FAD? Are more substrate linked to NAD than FAD? Yes. Also know that there is a unidirectional flow. If all substrate linked to NAD will pass 2H plus an electron right in one direction, not back and forth. Substrate linked to FAD will bypass which complex? But again, will flow unidirectionally, complex 1. Also, are they? So when you are saying the 2H plus an electron, whether from FAD to oxygen or from NAD to oxygen, how do they flow? Electrons participate within each complexes and hydrogen have that proton circuit. So proton circuit is then again different from the proton flow. There is a electron that flow participating with component and then there is a proton circuit. So how do electrons flow? Ion, Fe plus 2, Fe plus 3, Fe plus 2, Fe plus 3. That's why then you come to inhibitors. That's why do carbon monoxide cyanide have a specificity that one binds to Fe plus 2 and the other one binds to Fe plus 3. So which one binds to Fe plus 2? Carbon monoxide or cyanide? Fe plus 2. Carbon monoxide. And is the binding competitive? Is the binding competitive? Yes. So see, first you know normal flow. You then bring in inhibitors. Then you bring the specificity of the inhibitor. There is the mechanism of action. And lastly, please do the overall outcome. What is the effect of any inhibitor on oxygen consumption, ETC function, ATP synthesis? But when you say that carbon monoxide and cyanide binds to Fe plus 2, Fe plus 3, where is Fe plus 2, Fe plus 3 here? There's no iron sulfur. Iron sulfur is in 1, 2, and 3. There's no iron sulfur here. So how is this Fe plus 2? From cytochromes. Cytochromes are heme protein. Absolutely wonderful. So this is complex 1, 2, 3. This is known as cytochrome oxidase. So you know the flow then. Always don't be confused, please. It's always this. The first component oxidizes substrate. Next component reduces coenzymes. Then coenzymes get oxidized. Who will get reduced? When coenzyme gets oxidized, who will get reduced? When CoQ. When CoQ get oxidized, who will get reduced? Cytochrome C. When cytochrome C is oxidized, who gets reduced? Oxygen to water. Now this is normal. So you did everything. You did the how they flow. What gets oxidized, what gets reduced, right? So what is the function of complex one? To transfer H plus an electron from where to where? So they are also specific. What's the function of complex one? To transfer electron from, say, NADH plus H plus to CoQ. Because remember, that's the right thing to say. And not NAD to CoQ. NADH plus H plus to CoQ, right? Does that make sense? Are you understanding what I'm saying? Yes, that's the function of complex one. For example, then you bring inhibitor. For example, when complex 3 is inhibited, what will be CoQ state? Oxidized or reduced? When complex 3 is inhibited, reduced state. Then, okay, also, how will you first do what is the mechanism and outcome, then overall effect, unless you know the right inhibitor. So malonate is the inhibitor of malonate. Complex 2. So you understand how I'm trying to tell you the flow, what to go, and then why complex 2 is not a coupling site. Because is this increasing after oxidation? Is this increasing big time after oxidation? After oxidation, do you have lot of NADH and FADH2 in the matrix? More NADH? Yes. More NADH than FADH2. Now, inner mitochondrial membrane is impermeable to these H plus ions. So when they accumulate, where they flow? Through these complexes. Now, this proton circuit from matrix to where? What is this? Matrix to? This proton circuit is matrix to what? What is this? What is this? Intermembrane space and back to matrix. This is proton circuit. This is not proton flow. This is not electron flow. Electron flow by participating within the components ion surface center. This is the proton flow. Proton is flowing through which complex? 1, 2, 3, 4, or 5. Where is proton flowing? Through which complex? 5. Now this has a different inhibitor. So understanding also the first mechanistic normal different component also is complex 2 allowing this proton circuit. Is complex 2 mentioned here? No. So that's why complex 2 is not a coupling site. What do you mean by coupling? You're coupling oxidation with what? What do you mean by coupling? Coupling oxidation with phosphorylation. That is known as oxidative phosphorylation. So complex 2 is doing oxidation, but not phosphorylation, right? Then you have inhibitors. Then you bring in the inhibitors. But now tell me why inhibitors of electron transport chain and complex 5 have the same effect on ETC, oxygen consumption, and ATP synthesis. Why they both have the same effect? Either it's any electron transport chain inhibitor or complex 5 inhibitor. Why they have the same effect on ETC function, oxygen consumption, and ATP synthesis? Why? Because they are tightly coupled. They are tightly coupled. Yes, these two processes are tightly coupled. Any one of them affected by inhibitor, they will have the same outcome on ETC, function, oxygen consumption, and ATP synthesis. But look at uncouplers. They create a havoc. They abolish this proton circuit. And that is why, because of uncouplers, what gets dissociated now? What is dissociated now? Oxidation is dissociated from phosphorylation. So in the presence of uncouplers, that's why ETC does not know when to stop. Because who will stop the ETC? How will ETC function stop? Is there something that can stop the ETC when you have enough? What can stop the ETC? When you have enough ATP. But now uncouplers have done what? They have dissociated these two processes, which should have been tightly coupled. So what will happen? ETC will keep going in the presence of an uncoupler. What happens to ETC? On and on and on. And oxygen consumption also increased or decreased? Oxygen consumption also increased. But Is the ETC going on and on, trapping the useful energy into ATP still in the presence of uncoupler? No, and released as heat. ATP is the chief controller of ETC. When you uncouple oxidation from phosphorylation, ETC is like a spoiled child. Keeps going on and on without parental control. That parental control is ATP. Please, I just give example. There's nothing personal about that. That how you understand why there's a different effect on what? ETC function, oxygen consumption, and ATP synthesis with uncouplers compared to the other two. Why they have the same effect, I told you. Why ETC inhibitors and complex 5 have the same inhibitor? Because they are tightly, why they're tightly coupled. So that's why they have the same inhibitor. Why I explain uncoupler as total dissociation, that's why they have different effect on the overall effect. So see, first the normal flow. What are your components? Why are they looking different? And what complex helps in what H plus electron transfer from where to where. Then you bring in inhibitors. First, the right name. I asked you to. Very good. Somebody said malonate. Absolutely wonderful. Because if you don't know 2 is malonate, you can say it is inhibiting complex 5, first of all, right? Yes? If you don't know malonate is a complex 2 inhibitor, then if malonate is in question, you will pick it is an inhibitor of complex 5. So is malonate an inhibitor of complex 5? Is it true? No. So first you identify the right inhibitor. Then its mechanism of action, as I explained you in the previous slide. Then its overall outcome, right? Carbon monoxide and cyanide, very good. Somebody said carbon monoxide binds to Fe plus 2 of complex 4. Binding is reversible. Overcome by what? Carbon monoxide binding is overcome by giving what? Hyperbaric oxygen. High concentration, high pressure. Whereas cyanide binding is it covalent or competitive? Binding of cyanide to Fe plus 3 of complex 4 is covalent, cannot be reversed easily. So you need an antidote to act quick, which is amyl nitride. So see, we went a lot before this last overall outcome. Overall outcome we did before that, we identify, okay, this is the normal flow. Substrate linked to NAD, go where? FAD, go where? What are these components made up of? How do they differ? Is there another name? There are other names too. For example, complex 5 is also known as ATP synthase complex. Then what is complex 5 made up of? So is complex 1 made up of F0, F1 or complex 5? What is made up of F0? Complex 5. So first the normal. Then inhibitors, where do they act and their overall effect. This slide quick exactly summarize if you have a confusion on inhibitors. Everything is normal here. You add ATP and there is steep fall in oxygen concentration in the experiment. Very good. Everything is normal. This is what should happen. You have mitochondria, you have substrate, you have electron transport chain. Everything is good and ATP should start the ETC function. But now you added something in the experiment that there is a slow fall in oxygen concentration. That means there is reduced oxygen consumption. So who does reduced oxygen consumption? Which inhibitors? No, B could be which inhibitor? Which inhibitors can do reduced oxygen consumption? Can B be uncoupler? Can B be uncoupler? Yes, 1, 2, 3, 4 and oligomycin. B cannot be uncoupler because C is uncoupler because there's again a steep fall in oxygen concentration. That means there is an ETC is going on and on and on. There is increase in oxygen consumption and this is uncoupler. So how A is also causing the steep fall in oxygen concentration or increase in oxygen consumption by ETC. C is also causing a steep fall in oxygen concentration, increase in oxygen consumption by ETC. So they both are beneficial. Is adding A and adding C both was beneficial? How are they different? Which cannot be extrapolated from this graph. How are they different? Where are you seeing ATP synthesis after adding A or after adding C? When do you see A? That's the difference. After adding A, there's ADP synthesis, which you will not have after adding C. Does that make sense, right? Remember that uncouplers, though ETC is going on and on, still the energy in the presence of uncoupler is not being trapped as where C was, energy released is not being trapped in the presence of uncoupler as still ATP. It's not like it's beneficial. Uncouplers do not accompany ATP synthesis, though ETC is going on and on. Signal transduction, many are struggling with, but do not struggle, please. Do not stress. I'm telling you, you people have done enough. You are a good group, actually asking a lot of questions and being interactive. Just don't miss the flow. Be mechanist, be basic before going to clinical. That doesn't mean clinical is you leave out. No, every lecture would be tested, some more than the other because of the complexity of their content. That's why I told you signal transduction, enzyme, and ETC would be tested more. Signaling. First, you do the signaling molecule, then you do the receptor they act on, and then the signal transduction mechanism. So don't miss the beat, right? You know the beat of the music, right? If you miss the beat of the music, what happens? What happens to the music? Does it look good? Will it be pleasant to hear? No, same for signal transduction. Once you do that, first distinguishing signaling, is it lipophilic or hydrophilic? So if it is lipophilic, though they use intracellular or extracellular receptors. If they are lipophilic, they use intracellular receptor, which are cytosolic and nuclear. You separate them. Lipophilic are steroid thyroid superfamily. But now extracellular receptor is what is stressing you. What is extracellular receptors also known as? They are also known as anyone? I hope you're following my pace. I'm just trying to address something and everything and then also open question and answer. Yes, are you following my pace? Everybody? Great. So yes, first you do signaling molecule, very good answers. You divide them into lipophilic, hydrophilic. Lipophilic, very well answered, use intracellular receptors, which are cytosolic and nuclear. And what are these lipophilic messengers? Steroid thyroid superfamily. Now this is stressing more people is when hydrophilic messengers use extracellular receptors, they are also known as, or extracellular receptors also known as. Is there other name? There are other names in this exam, so you should know the other names. So what's the other name for extracellular receptors? Anyone? Are they also known as something? Plasma membrane receptor. Absolutely wonderful. Then you divide them into three categories. First, heptahelical. Put them out of the way because heptahelical is associated with trimeric G protein or monomeric G protein. So you do a trimeric G protein. Since they are more, first finish them. When you say trimeric G protein, it's GS alpha, GI alpha, and GQ alpha. I'm finishing heptahelical first, okay? So what did you do? You did the ligand, lipophilic, hydrophilic. Lipophilic, intracellular, cytosolic, nuclear. Example lipophilic, anyone? Example lipophilic, anyone? Steroid thyroid superfamily, yes, anything that comes under the blanket. Then hydrophilic messenger, extracellular receptor, also known as plasma membrane receptors. How many types you have to do? First two heptahelical, because they are associated with trimeric G protein, which is GS, GI, and GQ. When you do GI, I will tell you now in the next slides, first complete GI, the whole cascade, complete GS, complete GQ. Once you have done that, I'll tell you how to do it. Now you go to the tyrosine kinase receptor, put them in one blanket. Tyrosine kinase first do growth factor. So growth factor, the tyrosine kinase is already a dimer or they have intrinsic tyrosine kinase activity. That means the receptor is a monomer and dimerizes after growth factor binds. Is that, yes. No, it's not already a dimer. Already a dimer is insulin, yes. Growth factor, tyrosine kinase is monomer, dimerizes when growth factor binds. Then finish this tyrosine kinase. First, you finish heptahelical, then you go to tyrosine kinase. First, you do growth factor tyrosine kinase, So when you want to complete growth factor tyrosine kinase, I'll show you how to complete, but I'm asking you, when you're doing growth factor tyrosine kinase, is it activation of protein kinase A or activation of RAS then? If growth factor is RAS, so see, then putting the right cascade, I will tell you how to complete. Then you go to insulin, not these two. First, you go to insulin, which is a tyrosine kinase model. Insulin, then complete the insulin mechanism. Insulin docks different protein. So is insulin using RAS? Does insulin use RAS? Yes, it can by docking GRB2 protein. So also understanding that then insulin can have same mechanism as growth factor, because insulin has a growth factor like activity. So first identifying each pathway individually and then finding the commonality too. Is there any common connection? So that will help you. Then associated tyrosine kinase, I told you not to do in detail. Serine threonine kinase, I told you not to do in detail, right? So have you followed the focus, right? Everybody is good? Yes? You do plasma membrane, you do heptahelical, you do GSGR, complete the cascade. Then you go to tyrosine kinase. First, you do growth factor tyrosine kinase, then you do insulin tyrosine kinase model. I did not mention in my lecture to do JAK-STAT tyrosine kinase in detail or serine threonine kinase in detail. Let's go now next. How do you complete it? Now, this is using plasma membrane or intracellular receptors, steroid thyroid superfamily. It's using intracellular receptors. So if it will steroid thyroid hormones use heptahelical receptors? No. So who will use heptahelical receptor? So identify the right ligand, the right receptor, and the right signal transduction mechanism. Steroid thyroid family use intracellular receptor, act at the level of gene. Acting at the level of gene is long-term or short-term regulation. Acting at the level of gene is long-term or short-term? Long-term regulation, induction and repression. So then you connect your an overview with signal transduction. Please don't get lost in this big picture. This is for you to distinguish. For example, cyclic AMP, cyclic GMP. Are they proteins or they are second messenger? Cyclic AMP, cyclic GMP are proteins or second messenger. So that you should clearly know, right? Do you phosphorylate tyrosine residues for insulin? Do you phosphorylate tyrosine residues for insulin? Yes, so insulin has tyrosine kinase model, right? So specifically knowing what they are rather than memorizing this chart, because this will eventually come in. What do you mean by G protein coupled receptors? Which are they? The intracellular or the plasma membrane G protein coupled receptors? They are plasma membrane. And in plasma membrane, G protein coupled receptors are heptahelical or tyrosine kinase? G protein heptahelical. So see, you got to be specific when when you see them anywhere in your text, in your slides. Everything should flow in your mind. These 7 transmembrane domain. Am I talking about insulin, glucagon? Which one I'm talking about? If you see a 7 transmembrane domain, heptahelical receptors, am I talking about insulin or glucagon? I'm talking about glucagon. First, correct ligand, receptor, then which one, GS, GI, or GQ? I will tell you how to complete. Now tell me, is it GS or GQ? Which one of them, when active, will activate protein kinase C, GS or GQ? Which one? GQ. Exactly, not GS. GQ will activate protein kinase C. And when is GQ active? When bound to GTP or GDP? When bound to GTP or GDP? When is GQ active? GS, GI, GQ, when are they active? When are they active and bound to GTP? So again, first to identify the right ligand. For example, I said if it's heptahelical receptor, ligand is not insulin, it's glucagon, or it's epinephrine, but it's not insulin. If I say insulin and glucagon, heptahelical is not insulin, it's glucagon. Then are they recruiting GS, GI, or GQ? When they recruit GS, GI or activate GS, GI, when are they active? When they are bound to, when are GS, GI and GQ active? When they are bound to GTP. Also, when they are active, what protein they activate? For example, I asked you, GS or GQ, which will activate when active, will activate protein kinase C. GS or GQ? GQ. So see, you got to be specific in that flow. Please see, I've drawn these line diagrams. This is not AI. I drew them by my own hand. So I also have posted them on Truro site for everybody's benefit, if you benefit. But what I want to, whatever source you go, give me good grades, right? So ligand, receptor, what G protein they are using, second messenger, and lastly protein. For example, this was very good. When I said GQ, you said it's protein kinase. GQ was protein kinase. C, not protein kinase A, not protein kinase B, not protein kinase G, right? So let's do that. So after identifying ligands is hydrophilic or lipophilic, first two lipophilic, intracellular, cytosolic, nuclear, and work at the level of G, then go hydrophilic, plasma membrane, extracellular, then divide them into heptahelical, tyrosine kinase, heptahelical, trimeric G protein, do complete GS, GI, GQ, tyrosine kinase, do growth factor tyrosine kinase, and insulin tyrosine kinase. JAK-STAT tyrosine kinase, I didn't tell you to do in detail. Serine threonine kinase, not in details. For example, let's stay heptahelical receptor. So when I'm saying epinephrine, alpha-2 GI, what's happening to protein kinase A? Active or inactive? It is inactive. So then you have to complete the whole thing. What is missing in this cascade? What is missing in this line diagram? Anyone? What is missing in the line diagram? Is there anything missing? The protein kinases are missing. You add them. Very good summary slide, please. Glucagon receptors are absent in the muscle or liver. Glucagon receptors are absent in the muscle. So this is a good summary actually. But you have to club it with protein kinases. So trimeric G protein is not tyrosine kinase model. It's heptahelical. How many trimeric G protein you should know? GS, GI, and GQ. Either one of them, when active, they are bound to GS, GI, or GQ. When active, they are bound to GTP. Who else is bound to GTP and active? Who else is also active and bound to GTP? RAS. Absolutely wonderful. Either when you connect more within the lectures, or you connect between two lectures, better grades. You don't stop here. For example, are they heptahelical receptors here that I'm talking? Are they heptahelical? Is this heptahelical? This one is heptahelical? Yes, heptahelical. Remember epinephrine, glucagon, heptahelical receptor, trimeric G protein. So when you say GS, is it activating protein kinase A or adenylyl cyclase? What is GS activating? Protein kinases. So then there is a cascade. When it's GS, protein kinase A is not the next protein. Adenylyl cyclase is the next protein. So go stepwise. Also, please know that glucagon and epinephrine act by increasing or decreasing cyclic AMP. Glucagon and epinephrine act by increasing cyclic AMP. Yes, the G protein should be GS, but epinephrine and glucagon increase cyclic AMP. What does insulin do? Does insulin also increase cyclic AMP? No, it decreases cyclic AMP. Also, one confusion if you have, so the flow, there's a ligand, there's a receptor, there's a G protein, then there's a next protein, and then there is a second messenger, and then there is a final protein. So there is a cascade. And you very well answered with epinephrine and GI. Now, some people are confused between this red and this purple, red and blue, though they are opposite colors. So I chose red and blue. Please know that any stimulation has to be counteracted by inhibition. That's why they are there. GS and GI are, when GS function is over, GI will kick in. But still, their mechanism is different. So is GS activating PKA or GI? Their cascade is still is specific. So GS is activating pKa. Absolutely wonderful. But there is always a counter termination mechanism. GS has a GI. You know protein kinase A, right? If I say it's protein kinase B, if I say protein, this is protein kinase A, right? If I say protein kinase B, is it still epinephrine or it's insulin now? It's insulin now. So you quite trying to distinguish that. Then you get to this clinical cholera, pertussis. Now, please know that they are also acting on different G protein. But see, if you didn't know hydrophilic messenger, plasma membrane receptors, different type of G protein, their different outcomes. How would you do cholera? That's why when people jump to cholera pertussis, it will not make sense. If you know the cascade, cholera will come quick. For example, cholera modifies GS or GI. This is the modification, please. This is not active inactive. This is the modification by the toxin. So cholera modifies GS or GI? GS. If GS is modified, does cholera make GS active or inactive? Persistently active, persistently active. Now, this is the pathology you should know, but you know the basics. So if cholera is modifying, ADP modifying GS, making it persistently active, what will happen to cyclic AMP inside the cells? Increase. That you knew because you knew the basic, and that's responsible for rice water stool. So please cascade and fill them. For example, I asked you this, these protein kinase C, calmodulin kinase are not coming with GS, GI. What are they coming with? GQ. Also, there's a very good question on your online self-assessment. And some people say, are there enough questions? From one questions, you get 5 concept. Believe me, if you really spend time like that, one question needs 10 minutes because you will rule out the other answer. There was a question that lithium prevents IP3 degradation. So when lithium will prevent IP3 degradation, what will happen to IP3 inside the cell? I'm asking you specifically, will it become active or will its concentration increase? It don't say active, it's not an enzyme, it's a second messenger. So its concentration will increase. That's why first I asked you to distinguish the protein from the second messenger. So if lithium prevents degradation of IP3, IP3 concentration will increase, right? Now IP3 concentration, if it is increased, will it activate phospholipase C or protein kinase C? Protein kinase C. But if you answer phospholipase C, that means you don't know the cascade. So see, application is all based on your basic. If your basic is solid for this exam, application will come quick. IP3 is not, IP3 is activating protein kinase C, not phospholipase C. It's before the second messenger. PKC is after the second messenger. So you understand how this before and after is important for you to do application, right? So if lithium prevents degradation of IP3, then protein kinase C will be activated, not phospholipase C. So who is after IP3? Is it phospholipase C or protein kinase C? Who's after IP3? Protein kinase C. So cascade is not just important and for application also it's very important. Also, please see, I want to distinguish though, some students if are confused. Yes, IP3 moves calcium from ER to cell and that moves extracellular calcium inside. But both of them are still increasing calcium inside the cell. So in the end, I would say both kinases should be active. Protein kinase C or calmodulin kinase. Don't break your head which one is specific for that. Instead, if I'm asking about GS, will you talk about PKC and calmodulin kinase? GS. No, so that is more important than distinguishing PKC and calmodulin kinase. But if you want to do that, why not? More the merrier, right? So GQ is protein kinase C and calmodulin kinase. If you want to distinguish that, well and good. But what I do is I work for more students, not few students. So when I work for more students means what makes you pass? What makes you pass is you are not relating protein kinase C and calcium or calmodulin kinase to GS. Does that make sense? So do first what needs to know. Well, you can distinguish them. Very good. More grades, more grades. But don't flip-flop them. This is also a good summary slide, just like my line diagrams. They have also completed the protein kinase, right? So are you following? This is making you better with signal transduction? Like, great. Also, please see now, see GS, GI, GQ. First knowing GQ, first knowing that GS is adenyl cyclase, not GQ is important. Then you can bring in drugs. Masters do not have these drugs because they don't have CV. But all it's doing is first knowing the cascade. For example, albuterol is acting on GS. What will it do finally? If albuterol is GS, activating GS, acting through GS pathway, what will it finally increase? What will it increase finally with second messenger? With second messenger, okay, let me be cyclic AMP, not cyclic GMP, not IP3 and DAC. So it's still about basic signal transduction mechanism. I told you not to do these two in details, JAK-STAT and serine threonine kinase. Let's do growth factor. When growth factor signaling is on, remember, did you see this GRB2 with insulin? Because some students have a confusion here. Yes, GRB2 was insulin. So does insulin have a growth factor-like activity? Yes, it does. So when you see growth factor pathway, do you talk of PKB or RAS? Growth factor, so RAS. So just connecting the right protein and right mechanism. Does insulin also uses that? Absolutely. And is RAS also active like GS, GI, and GQ and bound to GTP? Is RAS also active? Yes, absolutely. Wonderful. Then you come to this cholera, pertussis. What is it modifying? So rice water stool is for cholera or pertussis? Rice water stool. GI is cholera. And we have not done neurofibromatosis. Actually, that will come with cancer genetics. But we have done this RAS always bound to GTP. What will that lead to? Always persistently bound to GTP cancer, right? But again, when you are doing RAS, know who is using RAS. That's growth factor. Can insulin also use RAS? Yes, look at this insulin. So insulin is a heptahelical or tyrosine kinase model? Insulin tyrosine kinase model. So when you are doing RAS, also know who can use GRB2. See here, is insulin also using GRB2? Like growth factor? Yes. So does insulin has a growth factor like activity? Why not? Insulin has a growth factor like it. But how is insulin different from growth factor? It has a tyrosine kinase model, which is already a dimer. But then insulin is diverse in role. Either it can dock GRB2, it can dock PI3 kinase, it can dock protein phosphatase. Growth factor is not doing that. So what's the similarity between growth factor and insulin then? What's the similarity between insulin and growth factor? Both of them use what? First of all, both of them use tyrosine kinase activity, right? Both of them use GRB2, but still insulin is different. How is insulin different? It docks different protein. So tell me, GRB2, phospholipase C, PI3 kinase, or protein phosphatase? Which protein insulin has to dock for growth factor-like activity? GRB2, phospholipase C, PI3 kinase, or protein phosphatase, GRB2. So it's also specific. The outcome is specific. The outcome of each docking protein is specific. Phospholipase C is not big in metabolism, but you know when I say phospholipase C, what is the protein kinase I'm talking? A, B, C, or G? Phospholipase C. What protein? A, B, C. Phospholipase C. When insulin uses phospholipase C protein, which protein kinase I'm talking? Yeah, you got to be specific. You got to be specific. Please read the question stem carefully. Are they talking about GQ? Are they talking about phospholipase C? Are they talking about adenylyl cyclase? Are they talking about GS, GI, GQ? Though insulin uses protein kinase B, that's in your head. But I'm asking you, if insulin uses phospholipase C, then it is protein kinase, not B. This protein, it is protein kinase C. Very good. Now, somebody is asking a question quickly, although I'm almost finishing. Yes. Let me read the question. Give me a second if I can go up. GI inhibit. Okay, keep the question to the end. No worries. I will address that. So insulin uses tyrosine kinase like growth factor, but its tyrosine kinase model is different, already a dimer. Insulin can dock different protein which growth factor cannot. That's why insulin has different function. When it docks these protein, they have different outcomes. So if insulin has to do growth factor like activity, it will not dock phospholipase C. It will not dock PI3 kinase. It will dock GRB2. And when insulin docks GRB2, then you go to the RAS pathway and complete it. Does that make sense? So this is how I'm differentiating. So to insulin docks different proteins for different outcome. Is IP3 same as PI3? Is IP3 same as PI3? Which one is the second messenger, PI3 or IP3? IP3 is the second messenger, so you have to be so specific, please. So protein kinase, we don't jump to protein kinase B. If insulin is using phospholipase C, it is protein kinase. If insulin uses phospholipase C, then it should be protein kinase C. If insulin is using GLUT4, then it is, if insulin is upregulating GLUT4, then it is protein kinase B. So this A, B, C, G, this GS, GI, GQ, pay attention on that. This ligand, pay attention on that. Pay attention on the ligand. Pay attention on GS, GI, GQ. Pay attention, is phospholipase C first or protein kinase C first? Which one is first? Phospholipase C or protein kinase C? Which comes first in GQ pathway? Phospholipase C. And which one is at the insulin GLUT4 is protein kinase C or B? Insulin GLUT4. Insulin GLUT4 is protein kinase B. So see, they are specific. Just pay attention on them and you will be fine. If you, and there's still time, believe me, a lot of things you can do in the last day is seeing things which are being said are focus. If you repeat them, you can still do it, right? Every signal transduction mechanism, please, has a termination. Please know that. For GS, it is GI, and similarly, there for others. This is also a good way to refreshing everything. Please don't do the stimulus. They will come in foundation 2. The stimulus, this first stimulus, will come in foundation 2. Though you know very well, after meals, what do you release? What hormone you release after meals? Insulin. And then insulin does the action. People are stressing with, nitric oxide pathways. Sildenafil will come with Dr. Binstock in HMP shunt. But yes, this is not protein kinase B, C, but nitric oxide is protein kinase what? G. Absolutely wonderful. Now somebody had questions. And I just wanted to give a quick recap, and I hope this helped everyone. I wish you all good luck. I'm sure you all will do well. Keep this focus in your mind. Great.